Okay, so the project that I did this summer was 3D tumor spheroid generation using a droplet microfluidic device. And the overall goal of this project was to develop a droplet microfluidic trapping array to generate, interrogate, and recover 3D spheroids. But the goal of my specific research within this study was to create a system to deliver nutrients to culturing cells. So we used breast cancer in this study because breast cancer affects 1 in 8 females, which is a shockingly large rate compared that the second highest cancer has a 1 in 17 prediction of getting it. A study by the American Cancer Society estimated that of the 480 cases that would be diagnosed in Louisiana, 3,900 of them would be attributed to female breast cancer. 3D tumor spheroids is what we used to study in this experiment, and they serve as the best model for studying this because they create a very real environment that mimics what real tumors in the body face. A spheroid is an aggregate of cells that we grew on a hydrogel scaffold. And these are better for studying cancer drugs because they mimic real in-body conditions and how cancer drugs would really fight actual cells in the body. Other approaches to studying this involve 2D monolayers of cells that are often grown in petri dishes that do not exhibit real features of cancer tumors. In the figure that I have on the screen, it was taken from a research paper that was a study between drug testing on 2D monolayers and 3D cancer spheroids. And as you can see from the trend lines, the spheroids versus the monolayer reacted very differently. And in a real life scenario, this could be the difference between someone's treatment working versus it not working and them having further issues with breast cancer. So, a microfluidic device is a microenvironment where we can manipulate small quantities of fluid, or in this case, biological agents. We use this device to form and culture 3D spheroids. How we do that is we flow oil and cell-laden hydrogel into the device, as is indicated by the blue and green arrows, and then that reaches the droplet formation junction where oil pinches off our cell-laden hydrogel into droplets, as you can see by the animation playing on the screen in the upper left corner. These droplets then flow through the device before they settle into the overhead trapping array, as is seen in the figure next to our animation. The cells then grow in these droplets on our hydrogel scaffold. However, a problem that we face with this is that around 4 days after culturing in the device, our cells begin to suffer and die from lack of nutrients and cellular waste accumulation. The goal of my specific research was to make a cost-effective system of delivering nutrients to the cells growing in the device. So to do this, we used a computational model of our device in COMSOL Multiphysics to model the mass transfer capabilities of the device. The modeling that we did showed us how the flow needed to be for our system to work. We modeled the specific array in order to test out flow rates, trap spacing, and trap heights, because those are factors that could affect how our cells grow and release cellular waste. Modeling for this is still in process, so we don't have any final results yet on this aspect of the study. After simulating mass transfer in our computational device, I designed a gravity-driven flow trial. In previous research done by other groups in this line of research, a syringe pump would have been used to disperse media in the device. Media refers to a fluid that contains cellular nutrients and the ability to carry away cellular waste. However, syringe pumps, which were the replacement, which was something that I was trying to replace, are very expensive, often costing up to $5,000 per pump and take up a lot of space in a lab bench. I was able to create a system that was a low-cost replacement for this equipment using a test tube holder, syringes, and plastic tubing. In order for my trial to be a successful replacement, the system would need to be able to stand alone for 24 hours without human interaction and have a flow rate through the device of approximately 100 microliters an hour. The concept for my alternative flow device was that instead of being forced out of the syringe, media would flow downward through tubing in a gravity-induced flow. In the trials that I studied, I did a comparison between 2 methods of gravity flow. In method number 1, as is indicated on the left, two syringes, in this case, some combination of a closed system and a reservoir at the same height, flowed water into the device. That's indicated by the red box. Those were our reservoirs. The outflow, after water had flowed through the device, came out into a separate second Petri dish on the table, which is indicated by the blue box in that image. During this method, we experimented with different volumes of water, syringe heights, and submerging the device versus not submerging the device, which could have affected water and air leaking into and out of our device. Overall, after testing, this method proved to not be very successful. It was unpredictable, not consistent, and did not have the flow rate of 100 microliters an hour that we needed. The lowest flow rate that I observed in this device was 185 microliters an hour, which was far higher than we needed for it to be a standalone system for 24 hours. In method 2, as you can see on the right, one syringe flowed media into the device while a closed system was put into the aqueous inlet, or the second one down. Our reservoir is indicated by the red box in the image. A collector replaced the outflow Petri dish that we had had before, was placed at a secondary height about an inch below our reservoir. It collected the water as it flowed out of the device and then back up into the syringe. In this method, we specifically experimented with the height and size of the collector and reservoir. Overall, this method was very successful. It worked very well, was easily reproduced, and had a flow rate that was almost exactly on target, with the lowest flow rate I observed in this being 83 microliters an hour, which is well within the range that we were trying to accomplish. However, there are some limitations with these methods, being that water often leaks out of the tubing where it plugs into our device, and that sometimes we experience evaporation in the water that we're attempting to flow through it, which could have affected results that I observed. So in conclusion, method 2 has the best flow and success rates with on-target flow rates and very good standalone system for 24 hours. Method 1 was largely unreliable and had much higher flow rates than we needed, making it not a good fit for this study. And for our comparison of different variables that we studied, the gravity-induced flow was far more successful in 5 milliliter syringes than 10 milliliter syringes. Future research that I would like to do with this involves experimenting with further types of gravity-induced flow, which could form better ways to do this than what I first studied. I would also like to integrate it into real experiments where we are using cells in order to see whether it is really successful or not. Here are my acknowledgements, and my sources, where applicable, were placed below the images that they described. That concludes my presentation. If anyone has any questions, I can take them now.
All right, thank you, Margaret. So John, we can go and start with you, Dr. Flake. We've been doing one question per judge just to stay within the 10-minute period. So if Dr. Flake, you want to go first.
Yeah, thanks, Margaret. It's a good presentation. I'm trying to understand if the difference between method 1 and method 2. Method 2, it seems like you have a height difference to drive the flow. Is that correct? And then method 1, you used another method?
Yes, in method 1, our reservoir, or the syringe where the water was flowing out of, was above the table. Our device was on the table, and the outflow where the water came out of the device was also placed on the table, which was method 1. In method 2, our reservoir was placed upwards with it flowing down into the device, where water out of the device then flowed back upwards into another syringe that was placed about an inch lower than our reservoir.
Okay. Did any particular reason why you chose that height difference?
Not precisely. Pressure and gravity work differently at different heights, which was probably why we did that. It also was helping with our variables to see exactly what was being differenced.
Okay, thank you.
Thank you, Dr. Flake. Dr. Constant?
So in either setup, but we'll focus on method 2, and you have the slides, so we're there. How often did you have to recharge the reservoir? Let that be my first question.
Okay, in this system, we were designing something that could stand alone by itself for 24 hours. So approximately every 24 hours, we would come back in and refill our reservoir.
And so in that 24 hours with the syringes, how much did the level change in the syringe in red in 24 hours?
The goal was that almost all of the water would flow out of it at a rate of 100 microliters an hour.
Okay. And that would be, and how much would that 100 microliters an hour? So it's 2,400 microliters in a day. How much of the syringe is that? I can't do it in my head.
Both of our syringes pictured hold 5 milliliters, but they were both filled with 4 milliliters.
Okay. So a pretty good change in the head between the two over the 24-hour period.
Yes.
Okay.
All right, thank you, Dr. Constant. Boz, we have time for one quick question.
Okay, really quickly, good job using assertion evidence, and you sounded really polished giving your information. So really nice job even when you were kind of on script there. My question is one that I've asked a couple of times. Can you tell me in a simple sentence, and I got the whole idea of the breast cancer and all of that, the larger motivation, but can you tell me in a straightforward sentence what you did and why you did it?
What we did was we flowed water through plastic tubing into a small device that's essentially a piece of silicone with grooves in it. And the water flowed through the grooves into a collector. The goal of this experiment was to see how gravity and pressure would act on the water and how fast it would make it flow at different heights and syringe volumes.
And that matters for breast cancer research specifically because?
The research that we're doing is growing small 3D tumors that are breast cancer to provide better drug screenings. Whenever our cells are culturing the device, however, we face a problem where the cells die around 4 days into our culturing period. The goal of my research was to create a nutrient delivery system that would provide nutrients to our malnourished dying cells.
Thank you. I think I understand this better than any so far today.